This video covers question 66 from the biology section of the 2018 NSAA paper. This question focuses on Dolly the sheep, who we're told was born in 1996. She was unusual in that she had no biological father, and the diagram below shows how she was produced. So in the diagram we have two different sheep. We have a mammary gland cell donor and an egg cell donor. Now before we go any further, we should notice that the mammary gland cell and the egg cells differ in that the mammary gland is an example of a somatic cell, whereas the egg cell is a haploid gamete. Okay, so then the mammary gland cells are removed and cultured, and then have the nucleus removed and kept, whereas the egg cell, we take an egg cell, remove the nucleus, and get rid of it, and then we end up with an egg cell without a nucleus in it, and the nucleus from our cultured mammary gland cells. So this will be a diploid nucleus containing the full complement of DNA, identical to that of the mammary gland cell donor, and the nucleus is fused with this empty egg cell. So now what we have is essentially the same as a zygote that you would get from the fusion of a sperm cell's DNA with an egg cell's DNA. So within the egg cell, normally you have the fusion of the DNA from two gametes in order to produce a diploid zygote. But in this case, we've just taken a diploid nucleus and inserted it into the empty egg cell. So what we can then do is grow it in culture to end up with an embryo. Going further, we can implant it in the uterus of a surrogate mother, and then we end up with the development of Dolly the lamb, as obviously in utero the embryo forms into a newborn. So based on that, we're asked a few questions. Which of these processes is occurring in the diagram? Here are our four statements: genetic engineering, mitosis, meiosis, and differentiation. So let's start with genetic engineering. Or rather, cloning is not the same as genetic engineering. There's a distinction in that genetic engineering involves the introduction of a new gene into a genome, perhaps from another organism, perhaps from another species, and the manipulation of the genome to create an entirely new set of DNA. Whereas in this case, we're taking an existing nucleus with an existing set of DNA and just moving it into a new cell, a new host for it. Because by placing this nucleus in the new host cell of an egg cell, that will allow the development of an embryo. So this isn't genetic engineering, it's cloning. Moving on to mitosis. Mitosis naturally has to happen in the production of new cells in the blastocyst, or the embryo that we're seeing, that's shown here. And also there's the fact that we have cultured mammary gland cells, and all somatic cells replicate themselves via mitosis to create genetically identical offspring, or daughter cells. So mitosis is undergoing. Meiosis. Now meiosis can be found in this diagram in that we have an egg cell donor who produced this egg cell that we then used for the rest of the cloning process. So this step here, in which we go from our egg cell donor to getting an egg cell, involves meiosis, because meiosis produces all gametes, including ova. Differentiation. Well, since we have a clump of genetically identical cells here, in order for them to form different types of cells, for example, the epithelial cells and muscle cells and nervous tissue and all the different types of tissue that you find within a fully grown newborn lamb, you're going to need those cells to differentiate. And that's through the selective expression of different parts of the genome in different tissues. And that's what differentiation is. So we can see that 2, 3, and 4 are the processes that undergo when you're cloning, and that gives us our answer, which is option E.